Good morning, students. Our today's topic is Chapter 11 Biotechnology Principles and Processes. So, first, what is biotechnology? Biotechnology deals with techniques of using live organisms or enzymes from organisms to produce products and processes useful to humans. So, in this technique, organism or the enzymes from the organisms are used to produce some products or processes which are useful to humans. However, it is used in a restricted sense today to refer to such of those processes which we use genetically modified organisms to achieve the same on a larger scale. Furthermore, other processes techniques are also included under biotechnology. For example, in vitro fertilization leading to a test tube baby, synthesizing a gene and using it, developing a DNA vaccine, or correcting a defective gene are all part of biotechnology. So, all these are included in biotechnology. According to European Federation of Biotechnology, the definition is as follows: the integration of natural science and organisms, cells, parts, and molecular analogues for products and services. This is the definition given by the European Federation of Biotechnology. Now, what are the principles of biotechnology? Among many, the two core techniques that enabled birth of modern biotechnology are genetic engineering and maintenance of sterile conditions. So, first, genetic engineering. This is a technique to alter the chemistry of genetic material, that may be DNA or RNA. So, in genetic engineering, some changes is produced in DNA or RNA, and to introduce these into the host organisms and thus change the phenotype of the organism. First, bringing some change in the DNA or RNA, then transferring that DNA or RNA into the host's organism, and then bringing some change, physical change, phenotypic, which can be seen from the outside, change in the host organism. So when some bioengineering occurs, that is entered inside the host body through the vector, and then some phenotypic change or character change appears in the host organism. Next is maintenance of sterile conditions. Ambience in chemical engineering processes to enable growth of only the desired microbes or eukaryotic cells in large quantities for the manufacture of biotechnological products like antibiotics, vaccine, enzymes, etc. Under second point, so creating such type of conditions by using chemical engineering, by producing with the help of certain microbes. So these are used in antibiotics, for producing antibiotics, vaccines, enzymes, etc. Under sterile conditions, we require the growth of only desired microbes which we want. We want the growth of that organism only. As we already know that sexual reproduction has more advantage over asexual reproduction. Under asexual reproduction, opportunities for variations and formulations of unique combinations of genetic setup, some of which may be beneficial to the organism as well as to the population, Under sexual reproduction, variations occurs, and these variations or new combinations are useful for the organisms and for the population also. Asexual reproduction preserves the genetic information while sexual reproduction permits variation. So, what is the advantage of asexual reproduction? Asexual reproduction preserves the genetic information whatever, because characters remains the same during asexual reproduction. No variations occur, means, or very less variations occur, and same parental character passes, so it brings less change, or the organism resembles to the parents. So under asexual reproduction, it preserves the genetic information. Same genetic information remains to the next progeny, while in sexual reproduction variations occurs. Traditional hybridization procedures used in plant and animal breedings very often lead to inclusion and multiplication of undesirable genes along with the desired genes. When under the natural conditions, sometimes some undesirable genes also comes along with the desired genes. The technique of genetic engineering, which include creation of recombinant DNA, use of gene cloning and gene transfer, overcome this limitation and allow us to isolate and introduce only one or a set of desired genes without introducing undesirable genes into the target organism. Under biotechnology or under recombinant DNA strategy, we take only the desired genes that is cloned and then transfer into the desired host. So in this way only desired characters are passed to the target organism under biotechnological techniques. Do you know the likely fate of a piece of DNA which is somehow transferred into an alien organism? Most likely, this piece of DNA would not be able to multiply itself in the progeny cells of the organism. When we transfer any DNA into the host, the DNA cannot self multiply in the host organism. But when it gets incorporated or integrated into the genome of the recipient, it may multiply and be inherited along with the host DNA. But when this introduced DNA combines with the genome, that is the DNA of the recipient means the host, then along with that host DNA and new one alien DNA then multiplies in the host cell. This is because the alien piece of DNA has become the part of a chromosome which has the ability to replicate. Because when the DNA replicate, now this new entered DNA also replicates with the host DNA. In a chromosome, there is a specific DNA sequence called the origin of replication, which is responsible for initiating replication. So in the DNA, when some point is there, some sequence is there from where the replication starts. This is known as origin of replication, ORI, origin of replication, from where the replication starts. Therefore, the multiplication of any alien piece of DNA in an organism, it needed to be a part of the chromosome which has a specific sequence known as origin of replication. So this new alien piece of DNA is introduced near the origin of replication so that this new DNA can also multiply with the host DNA. Thus an alien DNA is linked with the origin of replication so that the alien piece of DNA can replicate and multiply itself in the host organism. This can also be called as cloning or making multiple copies of any template DNA. This process is also known as cloning. Let us now focus on the first instance of construction of an artificial recombinant DNA molecule. The construction of the first recombinant DNA emerged from the possibility of linking a gene encoding antibiotic resistance with a native plasmid. Plasmid, what is plasmid? Plasmid is an autonomously replicating circular extrachromosomal DNA. In the bacteria, main DNA is present circular, and along with that some small circular bodies are present, that is known as plasmid. These are plasmids are the extra chromosomal DNA. In prokaryotes, one DNA molecule, one DNA strand is present. Along with that, some circular plasmids are present, which are extra chromosomal DNA. So, while constructing an artificial recombinant DNA molecule, a gene which codes for antibiotic resistance, was taken from a plasmid and introduced into the plasmid of Salmonella typhi. किसी एक बैक्टीरिया के प्लाज्मिड्स में जिसमें कि एंटीबायोटिक रेजिस्टेंस प्रॉपर्टीज़ हैं, उस जीन को लेकर साल्मोनेला टाइफिम्यूरम के प्लाज्मिड में उस जीन को इंसर्ट कर दिया. Stanley Cohen and Herbert Boyer, learn the names, accomplished this in 1972 by isolating the antibiotic resistance gene by cutting out a piece of DNA from a plasmid which was responsible for conferring antibiotic resistance. स्टैनले कोहेन और हर्बर्ट बोयर ने 1972 में एंटीबायोटिक रेजिस्टेंस जीन को कट करके उस पीस को साल्मोनेला टाइफी के डीएनए के प्लाज्मिड में इंसर्ट कर दिया, ऐड कर दिया. The cutting of DNA at specific location become possible with the discovery of so-called molecular scissors, that is the restriction enzymes. Restriction enzymes are known as molecular scissors. So they can cut the DNA at particular locations. So they are known as molecular scissors because they can cut the molecules from in between. The cut piece of DNA was then linked with the plasmid DNA. The piece which was cut then introduced into the another plasmid. Just remember, same molecular scissor, that is same restriction enzyme is used for cutting both the DNAs. One from where the DNA is taken and other in the host where DNA has to be introduced. Same मॉलिक्यूलर सीजर या सेम रिस्ट्रिक्शन एंजाइम यूज़ करेंगे जिस प्लाज्मिड से हमें उसको जीन ऑफ इंटरेस्ट लेना है उसके लिए, और जिसमें ऐड करना है उसमें जिसमें डालना है, उन दोनों में सेम रिस्ट्रिक्शन एंजाइम यूज़ करेंगे. दीज प्लाज्मिड डीएनए एक्ट एज वेक्टर्स टू ट्रांसफर द पीस ऑफ डीएनए अटैच्ड टू इट. So by using certain plasmid DNA which act as a vector, these DNAs are transferred to the host cell. A plasmid can be used as a vector to deliver an alien piece of DNA into the host cell. So here also plasmid can be used to transfer the genes. The linking of antibiotic resistance gene with the plasmid vector became possible with the enzyme ligase. So cutting is done by restriction enzyme and then joining. Then for joining that molecule something is required. For that also one enzyme is there known as DNA ligase. So ligase joins the DNA and restriction enzyme cuts the DNA. So DNA ligase which act as on cut DNA molecule joins their ends. DNA ligase joins the DNA ends. This makes a new combination of circular autonomously replicating DNA creating in vitro and is known as recombinant DNA. So now old plasmid with new gene of interest, this total is known as recombinant DNA. Means नया डीएनए, एक डीएनए जिसमें नया जीन ऑफ इंटरेस्ट आ गया, उसको बोलेंगे नया रिकॉम्बिनेंट डीएनए बन गया. When this DNA is transferred into E. coli bacteria closely related to Salmonella, E. coli resembles to Salmonella in many characters. It could replicate using the new host DNA polymerase enzyme and make multiple copies. Now, this new recombinant plasmid, when enters into E. coli, then it can also replicate and form many copies. The ability to multiply copies of antibiotic resistance gene in E. coli was called cloning, because making many copy is known as cloning of antibiotic resistance gene in E. coli. You can hence infer that there are three basic steps in genetically modifying an organism. First is identification of DNA with desired gene. First we will see which gene we require, which is our gene of interest, first point. Second is then introducing this identified DNA into the host cell, transferring this into the DNA into the host cell. Third is maintenance of introduced DNA in the host and transfer of the DNA to its progeny. And after that we will see that recombinant DNA which enters into the host cell grow properly under the proper conditions and give the result into the next progeny. तो मेन स्टेप्स क्या हैं जेनेटिकली मॉडिफाइंग एन ऑर्गेनिज्म में? पहले उसका डीएनए को आइडेंटिफाई करो, फिर डीएनए को होस्ट सेल में ट्रांसफ़र करो, और फिर ये देखना है कि वो होस्ट सेल में जाकर डिज़ायर्ड रिजल्ट ला रहा है, जो हम फिनोटाइप चाह रहे हैं वो फिनोटाइप अपीयर हो रहा है. सो दीज आर द मेन फीचर्स, बेसिक स्टेप्स for generation of the genetically modified organism. What is genetically modified organism? An organism in which we have added the gene of interest, जो हमने मॉडिफाई करके जीन बनाया था और जिस होस्ट में डाल दिया वो अब होस्ट बन गया genetically modified organism. So this we will continue afterwards. Thank